Hi everyone, I did my final project on fluoxetine, which is a drug that is a selective serotonin reuptake inhibitor. So just some background on the drug here. Fluoxetine is generic for Prozac, which you might know it as. It is primarily used as an antidepressant, but can also be used to treat other mental health issues such as OCD, bulimia, or panic disorder. And the way that fluoxetine works is that it increases serotonin. And serotonin is a neurotransmitter in the brain that will regulate emotions, moods, and thoughts. The way that fluoxetine is going to work is that it's going to prevent or inhibit serotonin transporters from removing those serotonin molecules from the synapse. And we'll see that in a lot more detail in just a little bit here. But the picture on the screen just shows a few variations of different fluoxetine capsules or tablets. And just a brief overview, simplified overview of how fluoxetine is going to work. So normally serotonin will be released into the synapse here, and then that protein will recycle it back into the neuron. But if we have fluoxetine, which is an SSRI, selective serotonin reuptake inhibitor, it's going to prevent that protein from doing its job, and therefore the serotonin will be released, but it's not going to be recycled, so it's going to stay in that synapse for longer. Now, I want to talk about the structure of the sodium-dependent serotonin transporter. This is that protein that's job is to recycle the serotonin. So first, we're going to look at the primary structure here. And primary structures of proteins are made up of the sequences of amino acids. And here we can see the long sequence of amino acids that make up every sodium-dependent serotonin transporter protein. However, if we put all these amino acids together, we are going to start to see the secondary structure, which is regular repeating patterns, but it's going to be in regards to the main chain. So not the side chains that come from those amino acids, but the main backbone, and it's going to be done through hydrogen bonding. And we're going to see different types of folding. In the serotonin transporters, we're going to see two different kinds. First, we'll see beta sheets, which we see on the left, and then there will also be alpha helices, and these again are determined by the hydrogen bonding. Now, if we put all this together, we will get our tertiary structure. This is going to be the overall folding pattern, where those secondary structures are relative to each other. So again, alpha helices and beta sheets. And this will also be determined by side chain interactions. So each of those amino acids has its own side chain, and those are going to interact with each other. So that unique sequence of amino acids will give us a very unique structure for the protein, which is going to be important for it to be able to do its own unique job. Next, just to take a quick look at the structure of fluoxetine itself. Again, this is going to be an inhibitor of those proteins we saw in the last few slides. It is going to do this through uncompetitive inhibition, so it's not going to bind directly where serotonin would usually bind on those proteins. It's going to bind elsewhere, but affect how that serotonin can interact with the protein. So it's pretty much going to block the serotonin from being able to bind to the protein and be recycled. The exact mechanism of how the fluoxetine binds is kind of unknown. It's still a source of research that's being done today still. But there is one theory that there's probably some sort of a halogen binding pocket in the protein. So we see on our fluoxetine molecule here that there's these three fluorines. These are halogen atoms, and they're actually very polar. So those fluorines could interact with other polar amino acids, or they could interact with, say, positively charged ones. And so that might be one way that it binds. Now we're going to go a little bit more in depth on the mechanism here. So just to let you know how this picture's set up, we have our first neuron that has our transporter protein, and then this is going to be our receiving neuron with our serotonin receptors, and then serotonin here in the synapse. And like I said, the location is that synapse, which is the area between our two neurons, so between the blue and orange neuron. And then again, here's just our main components of what's going on here. Now, normally we're going to have our protein, again, alpha helices, beta sheets, our overall structure. That is going to take those serotonin molecules that were in the synapse, and it's going to recycle them or reuptake them into the original neuron. And that's going to allow us to release those again later on, so we don't have to make more serotonin. However, this leaves very little serotonin in the synapse, and therefore very little serotonin that can bind to those receptors. So here we see only 2 out of the 5 serotonin receptors are bound to serotonin. Therefore, we're not going to get as many of like those mood stabilizing effects that serotonin has. However, if we introduce our fluoxetine molecule here, we see it's going to bind to that serotonin transporter, again, just showing its structure here. And what that's going to do is it will prevent the serotonin that's in the synapse from being recycled. So instead of having that serotonin go up into the original neuron, more of it is going to be available to interact with those receptors. So here we see instead of 2, we have 5 of our receptors bound to that serotonin, and therefore we can see more of the effects of serotonin. Now, why is this an important drug? So we saw that it increases serotonin levels, so less reuptake means more serotonin in the synapse, and therefore more serotonin binding to its receptors. This is going to have a mood stabilizing effect. So people taking fluoxetine will be happier, calmer, more focused, etc. And here we can see actual evidence of this. On the left, we see a depressed brain. The yellow areas are where there's brain activity. So you see in the depressed brain, very little of that brain is being activated. A lot of it is not. However, if we look at the not depressed brain, there's a lot more brain activity. So these drugs, these antidepressant drugs, can help someone that has a depressed brain go to this not depressed brain, and that's a pretty big deal. That mood stabilizing can be a game changer. So that's the importance of this. I hope you guys enjoyed, and here's just like my citations in case you want to go check out more about it. Thanks.